In every single second, inside of living cells, thousands of chemical reactions are taking place. In most biochemical reactions, enzymes are needed to carry out the reactions. Like for the breakdown of sucrose, sucrase enzyme acts and makes the reaction easy and faster. Enzymes act as a biological catalyst and increase the efficiency and speed of the reaction. Without these enzymes, the reaction would proceed at a very low speed, making life impossible. Enzymes increase the efficiency of reaction by decreasing activation energy. By this diagram, let's try to recognize the importance of enzyme. The chemical reaction which is occurred without enzymes need more energy to activate. If more activation energy is required, it lowers the speed of reaction. Same chemical reaction in the presence of enzyme is completed in minimum energy because enzyme decrease the large amount of activation energy and speed up the reaction. Enzymes are specific for each type of reactions and perform their specific functions. For example, amylase and lipase enzymes both help in digestion, but they perform different functions. Amylase enzyme break down the starch molecule into sugars, and lipase enzyme break down the fats and oils into fatty acids and glycerol. Amylase cannot break down the fats, and lipase cannot break down starch molecules. Enzymes is a protein which is composed of hundreds of amino acids. These amino acids join together and coil upon themselves to form a globular structure. Every enzyme has an active site where substrate is attached. Substrate means reactant. Reactant or substrate can be one or more than one. The condition in which substrate attach with enzyme is called enzyme-substrate complex. When reaction completed or reactant convert into product, they separate it out from the enzyme. In all this process, enzyme only act as a catalyst and do not affect the nature and properties of end product. Some enzymes cannot increase the efficiency of reaction alone. Enzymes require a non-protein substance for their proper functioning. Such non-protein substance is called cofactor. Cofactor act as a bridge between the substrate and the enzyme. They sit an active site of enzyme and aid in recognizing, attracting of substrate and repelling of their products. They increase the efficiency of enzyme. Moreover, sometimes they provide chemical energy to enzyme which makes the reaction easier and faster. Vitamins and some metal ions are the examples of cofactor. If cofactor is loosely attached to the enzyme, it is known as coenzyme. If cofactor is covalently bonded to the enzyme, it is known as prosthetic group. An enzyme without its cofactor is called apoenzyme. An enzyme with its cofactor is called holoenzyme. Like cofactors are the helper of enzymes, although inhibitors deactivate the enzyme. Inhibitors are the chemical substances which slow down or completely stop the activity of enzymes. There are two types of inhibitors: irreversible inhibitors and reversible inhibitors. Reversible inhibitors are further divided into two types: competitive inhibitors and non-competitive inhibitors. Irreversible inhibitors occupy the active site by forming covalent bonds, or they may physically block the active site of enzyme. In the presence of irreversible inhibitors, it is impossible for the substrate to attach at active site of enzyme. They react with enzyme and destroy its globular structure and turns into a useless enzyme. Reversible inhibitors form weak or non-covalent bond with enzyme. Generally, like irreversible inhibitors, reversible inhibitors do not react with enzyme. That's why they can easily remove by different ways. Their effect can also be neutralized completely or partially by an increase in the concentration of the substrate. Competitive inhibitors are like substrates. They attach with the active site and deactivate the catalytic site of enzyme. Thus, substrates do not attach and products are not formed. Non-competitive inhibitors attach other than the active site of enzyme. They alter the structure of enzyme in such a way that if even genuine substrate binds the active site, catalysis fails to take place.